Okay, everybody, let's go ahead and get started with the review. I think we've given enough time to let people join in. So we're going to be starting with our week 6 review. This is going to be talking about chapters 9 and 25, of course. Then chapter 10, we're going to cover the most missed topics for those. And I'll take any questions if you guys have any at the end, as we usually like to do. So we'll start with chapter 9, which is our microbial nutrition chapter. We talked, oh, well, I guess it's going to cover it here. We talked about the different, okay, guys, you cannot jump up on me while I'm doing this. We talked about the different types of how they get their carbon versus their energy. Seriously, they're right up in my face. Sorry. I thought that they'd be calmer if I let them in here. Not at all. Okay. So the trophs. So if we're looking at these guys here, the autotrophs and the heterotrophs. Okay, so these guys are talking about basically carbon, whereas the photo and chemo, these guys are talking about energy sources. Try to keep them a little straight in your head because and the odds are I will give you a choice of which one, uses inorganic sources for carbon, then you would need to know that, the difference between what is using carbon and what is using energy and all that. So I might give you choices like your 4 choices could be heterotroph, autotroph, phototroph, and chemotroph. So it might not be obvious. So be aware of that. Anyways, we talked we got into saprobes a little bit. We've already mentioned growth factor. That shouldn't be a new topic for people, but that is something that is required in the diet for something that it can't make it itself. So it has to have it in its diet, like our vitamins. And then we have the saprobes that digest the dead things. We call that detritus or detritus, however you want to pronounce it. And I'll tell you that we will absolutely have a question about the saprobes on there. There's a question about the autotroph versus, phototroph type of carbon versus energy stuff as well. So yeah, I know that. We didn't get into too much about the halophiles and the thermophiles, which means it's not going to be on the test. We talked about diffusion, which is movement according to a gradient, right? Natural movement with the gradient. So going from the high concentration, moving to the low to equal things out. And it's pretty similar to what we were talking about, what at least some of us were talking about in chapter 10, where we're trying to describe why those hydrogen ions need to be taken care of at the end, as well as the electrons at the end by that oxygen coming in to create water in the electron transport chain. Well, if you have a pool and you have a little wall in the middle of the pool and you take the wall out, the water is going to be even all the way across. If you put a hole in the middle of that wall, the water is going to be even all the way across eventually. So you don't necessarily always want it to be even on each side, but that need for it to become even when you have more on one side than the other, that's our gradient. That's what we're talking about. So going from that high to the low to try to even things out. That's going to be what drives diffusion. And so osmosis is just diffusion of water molecules. Okay, and so our lipid bilayer membrane that makes up the cytoplasmic membrane of cells, as well as for the most part, if you have another second membrane on the outside, it should be made up of a lipid bilayer as well. You just might have different molecules stuck in that lipid bilayer. So anyways, lipid bilayers make up membranes, no matter what organism you are. That's my point. And they can transport water freely across, and maybe some smaller nonpolar molecules as well. But things like salt and larger molecules can't move across. So if you put a cell into a solution that's a little more salty, the salt can't move across. Only the water can, to compensate for the difference in the solute, that salt. Okay, so remember that. The solute is also the term for molecules that are dissolved into the solvent, the dissolver, which in our most of the time is going to be water. That ability of the membrane to only allow that water and smaller nonpolar molecules across, that's called selective permeability. And then we talked about the differences in concentrations of solute from inside of the cell versus outside of the cell. And that's going to be isotonic, which is equal inside and out, hypotonic, where the solute is higher on the inside, and the outside is hypo compared to the inside, right? So hypo meaning lower, tonic, so lower concentration of the thing compared to the inside, which will be higher. And then the hypertonic solution will be higher on the outside, lower on the inside, okay? And the movement of the water molecules across our membrane, because we're only ever going to talk about movement of the water molecules, not the other things. Never the other things, okay? When we're talking about isotonic, hypotonic, or hypertonic, it's only ever going to be the water moving. It's only osmosis. So when we're talking about those situations, again, we want to have equal amounts of water on both sides of the wall in the pool. That's what we really want. Physics tells us that. So to keep the solute equal on both sides of the membrane, basically what the cell is going to do is move water, because it can't move the other thing, to try to equal out the concentration. It would make sense if we were just dealing with a little bit on each side. But unfortunately, most of the time you're dealing with large concentrations on the outside of the cell. The cell's not going to be able to do a whole lot about it unless it's just a small change, right? So it's fine when it's small changes. Large changes can cause serious damage in the cell. If you have a lot of water and less solute, so if you're hypotonic, less of the whatever's in it, salt or whatever it is, more watery than the cell insides are, then what's going to happen is that water from the outside is going to come in and dilute what's inside of the cell to try to make it the same concentration. So that'll end up being swelling of the cell as we try to compensate for that. That can swell the cell to the point that it bursts. That's called lysis, right? If we have the hypertonic solution, so we have a very salty or very sugary solution, something where we have a lot of something dissolved compared to what's inside the cell. If it's a high concentration outside, then the water will try to water the water. The cell insides will try to water down what is outside of it. So move water out to the outside to try to make the concentrations equal, to make up for the difference there. So that loss of water in the cell will cause that cell to shrink quite heavily. And that can be damaging to the cell as well. And that can lead to something called plasmolysis. Or if you like to think of it as plasmolysis, because we're talking about the cytoplasm, basically we're losing all that water. So it's essentially, going to be kaput, if you will, not working the way that it's supposed to be. Hi, spaghetti. And yes, so that will shrink up and shrivel the cell. What bacteria can do to compensate for these things is basically essentially have a cell wall in general. So when we are dealing with the hypotonic and we're swelling up, that cell wall just keeps the shape so we don't overswell and burst. When we're shrinking down because we're losing our water in the hypertonic situation, then the cell wall can be something that the cell membrane is attached to strong enough that we aren't having too much shrinkage of that cell. We can maintain the shape enough to keep the cell working for the most part. All right, if you need to move things across your membrane that can't do it on their own, so anything that isn't water, basically, if you need to move larger molecules, salts or sugars or whatever like that, you can put just like pores in the membrane and they'll still move by diffusion. So still trying to equal out the sides of the water on the sides of the pool, right? They'll still move on their own according to the gradient. That's called facilitated diffusion. It's still diffusion, still operating by the laws of that gradient, but it's facilitated because we created a pore. So now it's not blocked by the membrane and its function, its selective permeability. Next, we have active transport, which is transporting things across the membrane against the gradient, or if we need a lot more of it transported all at once, it's not fast enough, things like that. So anytime that we have to do against that law of physics, where we're naturally moving our molecules across with the gradient from area of high to low, if we need to go the opposite direction or go faster, we need to put energy in. And anytime you're having energy in, hey, when you're active, you need energy too. So active transport is moving things by using energy for whatever reason. All right, now we have, we talked a little bit about temperature tolerance in the chapter. We did not talk about it in the exam, so congratulations. We did talk about symbiosis, though, and the different forms of symbiosis. Symbiosis is a term for when two organisms are basically, their lives depend on one another for survival, or at least one of them depends on another for the survival. So it's like life dependent. We don't just interact with one another in nature. Sometimes our lives depend on that interaction, okay? That's what symbiosis is about. My hand got shaky. Then we have commensalism. That's our first one that I have here. This is where one organism benefits, but the other is unaffected. So only one is benefiting here. With mutualism, both organisms are benefiting. So we had the plus one benefiting here. The other one's not affected at all. Mutualism, we had both of them are experiencing good experiences, like the gut bacteria in our gut. They are, getting food from our diet and getting safety from being in our body, and they're breaking down foods that we cannot break down on our own. This starts happening from infancy, by the way, and we'll talk more about this when we get into, I think it's when we get into chapter 23, the gut stuff. So anyways, I think that's when we talk about it. It might be sooner than that, but all right, then we have, it might be chapter 13, actually, now that I think about it. That was sad that I don't know, because I said the next chapter that we're going over. It might be chapter 13. I'm not sure. But yeah, we'll talk about where your biome comes from and things like that. But hey, it starts in the uterus, they found out recently, and then really gets cranking once you have been born and start having your first, going through the birth itself will be your first exposure. And then that's your biggest issue. And then after that, it's like things that you eat and encounter in your life, essentially. So when you have milk, first, whether it's breast milk or formula and later on just milk, you can't break down sugars in the milk without certain bacteria in your gut. And that comes from the breast milk itself. If you're having breast milk, it can come from probiotics or bacteria that could be added in with certain formula. So you should always have probiotics involved in your formula if you're giving your baby formula. Or you could have, otherwise you could have issues with, their poop and stuff like that. So, and their digestion. And things like that. So it helps with that gut health and ability to digest things that you wouldn't be able to do otherwise. That's mutualism. That's us benefiting from the bacteria in our gut and them benefiting from us. It's a win-win. So mutualism is our win-win. And then we have parasitism. Parasitism is probably the biggest concept to be aware of in our pathogen side of microbiology, of course, because all of our pathogens are parasites. One organism is benefiting, that would be our pathogen, the one that causes disease. Whatever microbe that could be the plague, the bacteria that causes the plague. It could be the virus that causes COVID. It could be a fungus that causes fungal meningitis. Whatever it is, that thing is benefiting at your expense. It's living and thriving, but you are suffering because of it being there. That's a parasite. So those are microbial aspects of parasitism, where I know we all are aware of the macro aspect of parasitism, where you can talk about ticks and fleas and lice and things like that already fall into that category. The microbes do too, when they're causing pathogenic effects, causing damage to you. Obligate parasites are terms for parasites that literally cannot survive outside of the host. We have some organisms that we would call opportunistic pathogens. So they're like normal They don't really affect you in any other way, but in certain situations, they can cause disease. So that would be things like normal mold in the environment, things like aspergillus. It's just around all the time, that mold is. But then when you have allergies or something, that inflammation creates essentially damage in the tissue that allows that mold to take hold and create infection. It doesn't normally do that though, right? That's an opportunity that it took to do that. It becomes parasitic when it does that, but it's not obligated. It does not have to be a parasite. Things like COVID cannot exist without being a parasite. So it is an obligate parasite. And we even learned that all viruses are obligate intracellular. They have to be inside of the cell using your machinery, parasites, right? So remember that term. This is back to that concept. Next, we have the biofilms. These are bacteria that communicate using the quorum sensing. Be sure I have where is it at? I have to figure it out. Here it is. Sorry. All right. We didn't talk too much about quorum sensing in this. We mentioned it previously. But yeah, quorum sensing is just whenever bacteria are going to be in a biofilm and their chemical outputs from reacting to their environment signals to the other bacteria nearby what's going on in the biofilm overall and lets them decide what to do in that biofilm. Should we keep growing or should we break off and do our own thing? That sort of stuff. So that's quorum sensing. A quorum is when you have a group that is communicating with one another. Whenever we do faculty senate, we come together. There's like 13 of us, and we'll come together and we have to have a certain percentage of people show up in order to have the meeting. Otherwise, it's not representative. And we call that achieving quorum. So it's the same concept. All right, so we also talked about the bacterial growth curve. This was something we didn't talk too much in the lecture, but know those 4 steps, okay? So the first one would be the lag. This is the first step. The lag phase is just you introducing the bacteria into a new environment. They're having to adjust and respond to the new environment, the temperature, the nutrients, things like that, and then start replicating their DNA. You can't make new cells without replicating DNA in the cell parts, like new cell membranes and things like that, going through the division cycles. So that takes time. That's the lag phase. You don't have true multiplication until you have those things taken care of. When you start having exponential or logarithmic growth in the culture, that's the next phase. That's this part here, log or exponential phase. So you have doubling of those microorganisms at the fastest rate, typically that species can manage. And then we have the stationary phase. This is basically where your growth is going to be slowing, and you might have introduction of some cell death, usually just because the cells are getting older, some of the nutrients are getting taken up some, but you're still managing to have a steady, I would say, it'll flatten out because your growth and your death or lack of growth are going to equal out in a way that you flatten out. Then we have the death phase where predominantly death is going to be occurring. We have a decrease in the population of living cells. And this is occurring because of toxic products that come just from waste, from, using up nutrients, or from, products that are made from cells dying around you and releasing enzymes and things like that can be damaging. Loss of the nutrients in the broth or the environment that you're living in. Maybe loss of any gases or anything that you might need, like if you needed oxygen, if you're an aerobe. So anyways, this was chapter 9. I want to summarize it pretty well because I know some of us didn't get to hear a lecture at all. And those that did, we had it sort of summarized. Next, we had chapter 25. Chapter 25 dealt with, look at this one. So this doesn't put it in this perspective. Chapter 25 are key concepts that we went over here. We briefly touched on the carbon cycle. I don't know how to help you anymore with the carbon cycle if you don't understand how carbon dioxide works. So basically, photosynthesis and us exhaling carbon dioxide and that cycle being dependent within one another. So then we have, we exhale carbon dioxide, the plants use it to fix it into organic molecules. And then when they fix it into the organic molecules, we can eat the plants, or the saprobes can break down the dead plants, or other microorganisms can be involved in eating the plants. Whatever it is, they get the organic molecules from them. And then we already know when you go through the Krebs cycle and you are putting your food, your glucose or other molecules somewhere in the intermediate stages of that, you're going to be breaking off some carbon dioxide and exhaling that. That's where it comes from. And so that's where that cycle will come into play. That's the carbon cycle within living things. And of course, you can have combustion in there, natural or man-made, and that'll contribute to that. Methane is also part of the carbon cycle. Methane is a more potent greenhouse gas, and it's typically going to be made by our methanogens, which are just microbes that make methane. It can be bacteria, but a lot more commonly, it's going to be archaea. And these can be bacteria like in the guts of animals. Whenever the animals are passing gas, that'll be methane derived from bacteria, or they could be bacteria. More commonly, it's going to be bacteria from the environment, like in swamps and things like that. All right, we touched a little bit on the nitrogen, cycle. Essentially, the role of nitrogen in soil and in biology overall is pretty important. You need nitrogen because your DNA has nitrogenous bases. You need nitrogen because your proteins are made of amino acids, and amino refers to the amino group, which is the NH2 group. That's nitrogen. So you need a lot of nitrogen in your diet. You are going to get your nitrogen not from the environment around you, right? You're getting your nitrogen from what you eat. And so you depend on things like plants to have DNA and protein that you can break down and use to make your own DNA and protein. Those are your nitrogen sources. There's other sources in your food that you can get nitrogen from as well, but those are biggies. And the plants get their nitrogen from the soil, and the soil doesn't have a naturally usable form of nitrogen. We need bacteria to change it into usable forms. And there's certain stages that bacteria can go through in order to achieve that. And those mostly, we've talked about nitrogen fixation. That's not on this list here, but that's one of them. Ammonification, which is producing ammonia. Nitrification, which is NH3 changing to nitrite and then nitrite into nitrate. Bacteria are going to do that. And then denitrification, where nitrate is changed into N2, which is atmospheric nitrogen. So you don't need to know the definitions. You don't need to be able to tell me that denitrification is nitrate, NO3, changing into N2. Yeah, I don't need you to know that. This is just here so that you're aware of these different processes that can go on with bacteria. We'll need to touch on them a little bit more in the lab, but you're still not going to need to know the details then. I think it helps to kind of know that these are going on in the background whenever we do get to the lab, which isn't until the very, very, very end anyways. But anyway, so know that these are ways that these are things that happen. Know the terms, that there is ammonification, nitrification, denitrification. And the one that I left on here that we don't need to worry about how these things work, just we also have nitrogen fixation. Those are the four things that you can have going on with nitrogen. You don't need to know what they are, how they work, but that's the terms for them, okay? All right, we talked about the problems with plastics that are in the ocean. Contaminating the ocean. About 80% of the ocean's surface is covered by microplastics at this point, which is a serious issue. Microplastics and larger plastics like bottles and things like that can create reefs in the ocean that can allow microbes to grow on them, or other, colonies of things to grow on them where they shouldn't be, block out the light, use up nutrients in the area they shouldn't be, creating different nutrients in the area, changing the ecosystems completely. And like I mentioned in class, these islands can be as large as, there's one that is larger than Texas in the ocean. So that, you can imagine that creating a massive issue in the ocean life that lives beneath it. All right, water filtration and wastewater treatment. Basically, we will be going through processes of sedimentation, sludging, if we're talking about wastewater treatment, and that will be breaking down material by bacteria, and whatever is left is sludge, and sometimes it can't be broken down any further, so we would set that stuff aside. And what's left of the liquid, we can filter and treat with chemicals. And then if it was clean-ish water, from an aquifer or a river or something to begin with, and we treated it chemically and filtered and everything, we can turn that into drinking water. But if it was wastewater, actual wastewater sewage treatment, then we won't be usually using that for drinking water. We can use that for, watering golf courses or park areas, or we can return it back into the environment, into the rivers or lakes or streams or oceans or whatever, once it has reached a certain level of cleanliness that we find acceptable according to government regulations. We determine whether it's acceptable by looking for certain types of microbes in those water treatments. And we call those indicator bacteria. And the ones that we use most commonly are our fecal coliforms. Our coliform bacteria, if they are present in our water sample of our filtered water of whatever type, whether it came for your drinking water or from your wastewater, if you have fecal coliforms in there, it means that those bacteria got through the filtering process. If they got through, pathogens may have gotten through too, whether you see the pathogens or not. Those obvious true pathogens like cholera, like dysentery E. coli, or Shigella and things like these guys, we don't want them to be able to get through. Even if we don't see them in our tests, we don't want them to be able to get through. So we need to be sure that other things like them can't get through. The other things like them are our coliform bacteria. These indicate that susceptibility. Okay, so we use microbes for food. Obviously, they're great for bread, beer, wine, and cheese and yogurt and stuff like that. And then we looked at the end of this chapter, it talked about the One Health concept, which I'm not particularly, I don't think it needs to have a whole section dedicated to itself, but it talks about the effects of society and climate on disease and persistence and reemergence of certain diseases as a result of climate change that may be due to our acts. This can affect mosquito populations, and we kind of already know that. As we have global warming, we have increase of mosquito populations pushing further north. And that means that those mosquito-borne diseases that were associated south of us, things like dengue fever, which is a hemorrhagic fever, those things are pushing north as well, which is why we're seeing these now in areas like Texas and Florida. We didn't usually ever see these at all, ever. We're also seeing malaria in the United States occasionally in little patches for the similar reason. We never, ever saw these in America before. And that's due to the change of our climate. So we'll also encounter new pathogens that may not have, we as humans may never have seen. This can be due to that climate change or due to urbanization. So moving into areas that were previously, woodland or something like that, and now being exposed to these diseases. So the effects that we have on our environment that has led to climate change, or just other effects on our climate, like buildup of plastics or overforesting or whatever it is, that can affect our health because it's affecting the health of the ecosystem. And that's what One Health is talking about, taking care of the planet as well as other organisms on the planet and not just thinking about ourselves. All right, chapter 10 dealt with metabolism. It's supposed to focus on microbial metabolism, but we kind of just touched on metabolism in general. Remember, metabolism is all of the chemical reactions in a cell that can be broken down into catabolism and anabolism. So catabolism, cats are going to break things apart, and anabolism, like anabolic steroids, we're building things, we're making bonds. We introduced the concept of enzymes. These are just organic catalysts. They're usually proteins that are going to help lower activation energy, which is the energy needed for a reaction to go. They bind onto something called the substrate. That's the thing they're acting on. Now, the enzyme itself is not going to be used up or changed. So we can use it over and over and over again on substrate after substrate after substrate. Okay. You have one enzyme that acts on one substrate involved in one reaction. So that enzyme doesn't have multiple jobs. So like DNA polymerase, it's only DNA polymerase. It's not polymerase that does DNA and RNA, right? You have one for each of those. That's how the enzymes are going to be set up. The thing they act on is called the substrate, and then it will make products at the end as a result of this interaction. and that could be just the substrate getting a new group attached to it or taken off of it. It could be simple things like that. Some enzymes are just only simple protein. They're not got any other parts to them. But if they are conjugated enzymes, then those conjugated enzymes, the whole working part is called the holoenzyme. The protein part is called the apoenzyme. And the part that the apoenzyme needs in order to work, the part that it binds with, is called the cofactor. The cofactor can be inorganic. It's still just called a cofactor. If that's the case, that's usually metal ions like iron or zinc or something. Or if it's an organic cofactor, it can be called a coenzyme. So remember, coenzymes are still cofactors. They're just an organic cofactor. A lot of times those coenzymes are going to be vitamins, but we even talk about later on in the chapter about how we have electron carriers, NAD and FAD. Those guys are coenzymes. They are coenzymes. So therefore they are organic molecules that are cofactors that will bind with enzymes later on when they interact with things in the electron transport chain. And that's how they're going to do their business. Okay. We learned about the enzyme. Oh, yeah. So the active site is where that substrate binds, wherever that enzyme interacts with your substrate. Okay. So our different enzyme classes that we talked about were oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. If anything, I would hope that if we went through the DNA replication chapter and the genetic engineering chapter, that we talked about DNA ligases, and you got it, that the DNA ligase sealed those bonds, so made bonds between the nucleotides to make sure we had a continuous strand, either with the Okazaki fragments or when we're doing genetic engineering with our little sticky ends or something like that. We have to seal it back up. That just means making new bonds between the nucleotides in those gaps. So we're creating bonds with those. So that, if you can remember that, that's going to be the ligase is create bonds. So if you're creating, you're always going to need ATP, always. All right. And if we're creating bonds, also this one's going to give off water. And it's also, so it needs ATP and it gives H2O. All right. The other, we don't care about these, right? We don't care about lyases. That's the double bond thing. We're not going to talk about isomerases. That's the changing between the shapes. So hydrolases, be aware of those. These guys are going to be breaking bonds because these are the other side. So if I told you that, okay, there's those 6 types, but you only need to know oxidoreductase, transferase, hydrolase, ligase. I would hope that you could break it down logically and remember that ligases are like DNA ligase, creating bonds. If they're creating bonds, they need energy, okay? And if you're putting 2 things together, they usually were attached to other things holding electrons to begin with. So you're going to have to get rid of those things. And in this case, it's going to be water. So we have water as a byproduct there when we're creating those bonds. However, the opposite side of that is the hydrolases. These guys are breaking bonds. And so if you were a ligase and your bond that you created gave off water, and this is the opposite, then obviously these are going to use water in order to kind of seal up those bonds that you just broke, the ends of them. Okay. So those are the opposites for one another. Transferases are literally just moving a group from one thing to another group to another thing. So that would be taking a phosphate group off of that molecule over there and sticking it onto ADP to make it ATP. All right. So just moving it from one thing to another. That's transferases, literally transferring. Oxidoreductases, though, that's like one of the more important concepts that goes on in this chapter. Oxidoreductases are dealing with electron transfer. So I'm going to circle that one really quick because I feel like transferase speaks for itself. This is electron transfer. Cool. All right, so our enzymes, you can denature our enzymes with heat or with pH change, usually extreme pH change. So lots of H plus ions, a higher H plus or proton concentration, Higher proton concentration is acidic, so it's a lower pH, right? Because remember, it's inverse there. And then if you're at basic pH towards 14, that means you have less H pluses. These extreme environments can affect how our hydrogen bonds work in our proteins with our amino acids interacting across from one another, creating those overall secondary and tertiary structures of our proteins, and even potentially the quaternary structures. We have multiple polypeptides found together in our structure. We need to maintain our hydrogen bonds in our proteins. So these things can disrupt that heat or pH. And when that happens, when we have that disruption, it can even be permanent. And so that disruption in general, where you can no longer function, is called denaturation. The natural state is that functioning state, and the denatured state is that state you achieve when you have messed up that functional structure. That can also just make that binding site completely useless as well. So it just needs to be enough of a change for that to happen. You don't have to completely mess up the entire thing down to like a straight line, but just messing up the binding site, the active site where the substrate binds, that's enough to denature an enzyme. All right, we can have, sorry, let me check this. Sorry, I got an email. We can have different ways to control our enzyme function. That could be controlling the enzyme at the enzyme level. And if we're doing that, then we're talking about our competitive or non-competitive inhibitors. Competitive inhibitors are inhibitors that are going to be binding at the active site. So that's where the substrate normally would bind, therefore blocking the substrate from being able to bind. If we have non-competitive inhibitors, it's usually the product of your reaction, and it's not being used up by the cell. So when it builds up, it can now interact with your enzyme, changing its shape somewhere out by binding somewhere else on the enzyme, anywhere else on the enzyme, changing its shape so that the active site is no longer structurally available for the substrate to bind in, effectively shutting it down. The difference between those two then would be competitive is talking about binding at the inhibitor, binding at the active site, whereas non-competitive is talking about the inhibitor binding anywhere else. Okay, that's what it's talking about. All right, you can also control enzyme production at the genetic level. This would still be considered inducing or repressing those genes. Keep in mind that if you're inducing or repressing at the genetic level, and you're only inducing or repressing one gene, that can happen in any organism at all that has genetic information. However, if you are inducing or repressing and it affects a whole group of genes down the line that are all associated with that one function, so several genes all at once, that's an operon, and that only works in bacteria. Okay. All right, so we talked about redox reactions. Leo says GER or oil rig, whatever you like. I have Leo says GER on this one, but we went over the other one in class. I'm sure you remember. Whatever works for you, you do need to know that losing electrons is being oxidized and gaining electrons is being reduced, because we follow that through in a lot of our steps in the next parts of what we're talking about in chapter 10. All of our redox reactions, you have to have something donating an electron and another thing accepting. You always have both, and they have to be accounted for at all times. Electrons aren't just going to disappear into the ether, like we've mentioned before. If you're putting an electron into a system or into a set of reactions, then it has to go somewhere at the end. It has to end up with another molecule as like a finished product. When we send things through the electron transport chain in aerobic respiration, our electron that we send through is going to end up finally, with our final electron acceptor of oxygen. In aerobic respiration, oxygen is our final electron acceptor. In anaerobic respiration, it is not oxygen, and that's all you need to know with that one, okay? So, ATP is our energy molecule. We should know about this one by now. The phosphates are what's holding, pushing each other apart and holding the energy because they're being forced to be bonded together. Aerobic respiration involves 3 major steps: glycolysis, Krebs, electron transport chain. Glycolysis, the main part of glycolysis is for, let me just go ahead and do this because we're going to do a little mini review of this so that you guys have that. All right, so with glycolysis, you will have glucose, and this is a 6-carbon, and this will be down to 2 of these 3-carbon pyruvic acids. Please try to remember pyruvic acid and pyruvate are essentially, for our practice and purposes, the same thing, okay? So, what are you doing, meatball? Right, so that's the main goal of glycolysis. In the meantime, you're also going to be giving off a little bit of ATP and a little bit of NADH. So I already mentioned NAD and FAD as our electron carrier coenzymes. So if it has that H on it, and that H is our carrier of our electron in our oxidoreductase reactions, so it's carrying an electron with it, and that's why it can create the bond and share that electron with that covalent bond to share that electron with NAD. So that's the whole reason that can happen, is because it's bringing an electron to the table. That's what we're interested in, is that electron carrier carrying that electron. So I'll even put this here to remind you guys, electron carrier. And this is reduced because this means it's carrying an H. So it's reduced. I don't know why that word looks weird to me right now, but it does. Anyways, so that's glycolysis and kind of what it does. Big picture, like glucose to the pyruvic acid. You do give off some of this other stuff. I would be aware that you give it off. I don't need to know how many. I don't need you to know the processes in between. All right, there is an in-between step. So here's our in-between prep step. We're going to take pyruvic acid and turn it into both of them, by the way, each one. So we'll have two carbon, something called acetyl-CoA. All right. So that's our in-between prep step. And when we do that, we notice that we've given off a carbon for each pyruvic acid. So for each pyruvic acid, we've lost a carbon. So that comes off in the form of CO2. That's the first CO2 you're going to give off. And you're also going to make, in this process, NADH. Again, here's another electron, right? So carrying an electron. So that's for each pyruvic acid. So now we've got our acetyl-CoA, or if we're looking at the 2 pyruvic acids, 2 acetyl-CoAs. And then this is going to go through the Krebs cycle. So this will be Krebs. All right. So acetyl-CoA comes in and binds with, it's like oxaloacetic acid or something like this, and oxalic acid, I believe. And that is, no, it's oxaloacetate, I remember. Yeah, oxaloacetic acid. It's going to come in and bind out. You don't need to know it. I'm just remembering for myself. It binds with something that's already come through the Krebs cycle. It's the leftover product of already had something else going through the Krebs cycle. And that something else happened to be another acetyl-CoA molecule, because it's this freaking cycle. So acetyl-CoA comes in, gets associated with a 4-carbon leftover something. And that 4-carbon plus the 2-carbon makes a 6-carbon. Great. That happens to be citric acid, by the way. It's also called the citric acid cycle. I'm not going to ever call it that on my tests, though. It's only ever going to be called the Krebs cycle. And anyways, then we're going to mess around with the bonds and how the hydrogens are associated with the molecules, and move things around, break bonds, release things, steal electrons, and move everything through until we come back to our final product of that oxaloacetate, as the byproduct of everything we've done, we could possibly do, combining the original acetyl-CoA with oxaloacetate to make citric acid, rearranging everything, done everything that we can in that cycle and coming back to that product so we can do it again and again and again. That's all the Krebs cycle is. So this is a big old cycle. We're going to put something in and then combine it with the waste of the previous and then go through the cycle and end up with the waste that's going to go with the next thing because now you're the waste from the previous. So that's how the cycle is going to work and why it's a cycle. The things that you give off in the Krebs cycle that are important, the big daddy important guys for this step are going to be the NADH and FADH2. These are our electron carrier coenzymes. It's really bad, but you can tell what it says. All right, so that's the main thing that we want out of the Krebs cycle. What else do we get from the Krebs cycle while we're shifting around all these bonds that we were able to do thanks to introducing the acetyl-CoA and combining it with the 4-carbon thing and shooting it through as citric acid? We're going to give off carbon dioxide because, hey, if I'm introducing this 2-carbon thing, binding it with a 4-carbon thing, and it makes a 6-carbon thing, and we're going to break it back down to a 4-carbon thing in the whole process in the middle, what the heck is happening to everything in the middle? So this is what's happening. You stole electrons in the form of hydrogens. We already saw this, right? Here's that. That's our electrons. These guys are reduced now because they gained electrons. Leo says grr, right? So they gained their electrons. And anyways, this Krebs cycle, you're also going to give off during that process more CO2 and just a little ATP. Okay. So now we can take all of these guys that we've made from all of these steps. We have, like I said, this is an in-between. It's not a true step, but there's something that happens there. So I want you guys to be aware kind of how that's working out. But what the whole goal of all of this is, so the whole goal of glycolysis and Krebs cycle together is to get to our electron carriers, make those guys. Glycolysis had to happen to give us pyruvate. Pyruvate had to happen to give us the acetyl-CoA that'll go into the Krebs cycle so we can move things around and steal off electrons. These are the things at the side that we kick off in the meantime, okay? So anything that comes off to the side in this image is stuff that we're kicking off in the middle of it all. It's not the big daddy goals. Stuff at the bottom in arrows that are going down are our big daddy goals. So for glycolysis, we went from glucose to our big daddy goal of pyruvic acid. For the Krebs cycle, we went from that pyruvic acid. Yes, we did go to acetyl-CoA in that intermediate step. If you want to call that the big daddy of the in-between step, that's fine. But the bigger idea here is that Krebs cycle is going to give us our big daddy goal of our electron carriers. Okay, so that's going to sum up what's really going on throughout the entire process leading up to the electron transport chain. Now, all of this stuff up above here that I've outlined up here, all of this and up, like everything up there, can go on in aerobic or anaerobic. Okay, if we're talking about glycolysis only, this glycolysis, this part can be aerobic, anaerobic, and fermenters. Okay, fermenters, just in case you couldn't tell from all of that, fermenters only go through glycolysis. Aerobic and anaerobic guys will go through glycolysis and Krebs the exact same way. Now, aerobic and anaerobic, they'll also go through these guys, I don't want to look at this. They also do go through, I'll put a little asterisk here. So, they do go through the electron transport chain. The difference is, of course, okay, I can't write below that, can I? Aerobic will have O2 as the final electron acceptor, and then anaerobic will have anything else, not O2. Okay. So now that we're going through the electron transport chain, we're going to shuttle our electrons through these molecules that we call cytochromes. All right, and then now that we've taken our electrons themselves off of the hydrogens are now H pluses. And we can take H pluses from other areas in the cycle as well, it turns out. But when those negative electrons excite those proteins as they go through systematically, they will move these H pluses out of the membrane. This is in the membrane, this is out. So then suddenly now, as we're doing that the whole way across, we get plus and plus, plus and plus building up there. So we have a positive charge outside, and the inside is, relatively speaking, negative. So now we've created this issue like a dam holding back water. And that dam is holding back H pluses, those protons, right? And they want to come back in to equal out the sides of the dam, the sides of the pool, right? To make the water equal on both sides of the wall. So that's what they want. That's what physics tells them to do. So they can only come through this little channel. And this channel is called ATP synthase. So ACE because it's an enzyme and we're synthesizing ATP. Go figure. As these pluses move through here, it drives ADP to become ATP and everything like that. So great. So we've moved electrons through. So we get buildup of electrons essentially at the end over here. Electrons, electrons, electrons. And as we're moving our H pluses through, we're going to get pluses and pluses and pluses hanging out on the outside. So we need to get rid of these because otherwise, if we don't, we won't have that gradient that's driving us anymore. Like the dam, if we let too much of the water out, now you don't have water to push through and make your energy anymore, right? The water's equal on both sides. You're not going to have diffusion moving that dam to make that energy anymore. So we have to get rid of them. So what we do with those excess, we take the H pluses, because these are H pluses, by the way. We take our H pluses with our electrons and then our water, our oxygen. We can split it up appropriately to make H2O. So our final electron acceptor, because it's the last thing accepting our leftover electrons, is oxygen. So now it is stealing off those electrons and doing so by reassociating them with those H pluses so they can share electrons as H2O in their covalent bonds. And so that's where, that's where we're using our oxygen up when you're breathing in. That's why you breathe in that oxygen. And that's why you exhale the carbon dioxide, because of these steps in here. So that is essentially respiration. Why is there so much barking? That's essentially respiration in a nutshell for aerobic and anaerobic. So anaerobic is the exact same. It just doesn't use oxygen. It just uses something else, anything else. I don't know what is going on there. Something. Anyways, so yeah, if you are fermenting, you just only go through glycolysis. So you stop. Your byproduct is pyruvic acid. You have this weird electron carrier that you had made up here at the very top, up here, this guy. you still have to get rid of that electron. It can't just sit like that for the rest of its life. So what you do to get rid of it is you recombine it back into pyruvic acid, and you make all sorts of products as a result of that. You'll make ethanol, or you can make lactic acid. You can make CO2 as a byproduct as well during this. And all of these things are important as far as making products that, like bread or cheese or beer or wine or, kombucha or yogurt, things like that. Okay, cool. All right, I think that was good for most of this, I hope. If you are, one other thing you're going to need to know for the exam, and we did talk about this, we just didn't focus on it like for a very long time, but that is in our little picture that we've learned about from this to begin with. We learned about that the electron transport chain was on the inner membrane of our bacteria. If you can see the image clearly in your mind, you'll remember that thick peptidoglycan purple guy on top of our membrane where our cytochromes were. So our cytoplasmic membrane or inner membrane for bacteria or archaea, this is where our electron transport chain, the aerobic respiration is going to be happening. So the glycolysis and Krebs can happen out in the cytoplasm of anything, any type of organism, but that electron transport chain. What? The electron transport chain molecules and the cytochromes, they will be either on that inner membrane or cytoplasmic membrane of the bacteria or archaea, or on the folds of the cristae, which are essentially the inner membrane of our mitochondria. Yeah, I hope I said all that right. I don't know. Some of the words are getting mixed in my head, so hopefully that made sense. So we kind of talked about our products. I'm not going to ask you about the exact numbers that are made, but I would definitely have paid attention to the fact that, oh, it doesn't let me go back to that thing I drew. All of that drawing that I did is gone now. Anyways, all of that stuff that we drew, we found out that we had in glycolysis very little ATP, only about 2 net for each glucose molecule, okay? So per each glucose, glycolysis, very little, Krebs, very little. And then the electron transport chain, most by far. Here we're talking about a difference of 2 ATP and 2 ATP and freaking 36 ATP or 34 ATP. All right, so most of your ATP is coming from the electron transport chain, whether you are bacteria or eukaryote. Most of it's coming from the electron transport chain if you're going through aerobic or anaerobic, typical anaerobic respiration. If you are fermenting, you're only going through glycolysis, so you're only getting that 2 from each glucose molecule, and that's it. So you can see how that's not very friendly for making new guys. Amphibolism is a term that's referring to the fact that you can go through glycolysis and make pyruvate, and then use that pyruvate to make nucleic acids or amino acids, or enter into other pathways that the cell might go into. Or you can take other things that you're eating that are not glucose or other sugars, things like lipids or things like proteins, and you can convert them into intermediates that can enter in at certain points in this whole pathway as well. So that's amphibolism, the ability to sort of exchange byproducts at any given point of any of these chemical reactions in the cell. That's what the term is about. We already talked about fermentation. Photosynthesis in the light cycle, we're basically making ATP. That's the goal. It's a very similar process to the electron transport chain, except we're exciting it with light instead of putting electrons in from breaking bonds. So light will excite chlorophyll or other pigments, create excited electrons essentially in the molecules of that chain, and we'll get a little bit of ATP. That ATP that happened in photosynthesis will then go into making into the light independent parts of photosynthesis. The light independent reactions use that little bit of ATP from the light reactions to fix carbon dioxide into 6-carbon glucose molecules. Now, if you think about the very little ATP making going on in that photosynthesis part, the light part, and then making enough ATP that we can power fixing carbon into glucose, and that can be a precursor to any of the other organic molecules that the plants are getting. Plants do not get their carbon molecules from anywhere else except for inorganic carbon. We already learned that in chapter 9, right? If they're autotrophs, then they're getting their carbon from inorganic carbon sources. That's carbon dioxide for plants, for example. So things like if they're getting, amino acids, or they're making their nucleic acids, or they're building new cell walls made of cellulose in their case, if it's plants, or whatever it is they have to make in their cell that's based in carbon, they have to fix it from carbon dioxide and build it from that glucose as that intermediate or other molecules along in the process. So great. So that's a lot of energy. How are we going to fix all of that carbon dioxide to make all of these molecules? Because certainly that ATP from the light reaction isn't enough to power every single thing in the cell. And you are absolutely right. The reason that we make glucose in those light independent reactions, the reason we use the ATP to make that glucose is so that we can shove that glucose right back into aerobic respiration, just like you and me. Y'all, plants have mitochondria. Yeah, they get what I'm saying here. Aerobic respiration. They use oxygen as their final electron acceptor. Aerobic respiration. They just happen when they're making their carbon molecules, because they have to make so much carbon molecules, they just happen to give off more oxygen than they need to use. So we think of them as being oxygen producers, when in fact, if you deprive plants of oxygen, they'll die too. So interesting, interesting the dichotomy we created there, but this is the truth. So that's how plants are going to get the energy to fix even more carbon dioxide to make more molecules in the cell, because they made glucose with that ATP they got from the sun. They can put that into our very heavy ATP output of the aerobic respiration in the mitochondria. Let me check this really quick, make sure that we're going to get to that in a second. I just want to be sure that some people here, I don't know if I have. I just don't want to miss anything like chats or whatever, because it doesn't alert me all the time on this way. So that's all I'm checking for. All right, so chapter 9's missed topics. Osmosis. Go figure that people wouldn't be getting things right about the tonic stuff. So we'll go back over this. Osmosis, movement of water by diffusion across a selectively permeable membrane from an area of high to an area of low. Okay, so that's talking about the water concentration. If you're talking about the solute concentration, that would be the opposite, right? Because the water is going to move to accommodate those concentrations. So keep in mind that we're trying to reach equilibrium of both on each side of the membrane, but we can only move the water. So the water is going to move from high to low water. So it's still diffusion, osmosis. So tonicity is referring to the concentration of the solute in relation to what's going on in the cell. This is usually dissolved in water. So an isotonic solution means that we're the same concentration of water and the same concentration of solute on both sides. So the water molecules will cross at an equal rate across the membrane. They don't ever stop. They're always going to be moving because science. All right, hypotonic solution. This is whenever the solute concentration outside is lower. So water will move into the cell to try to water down the cell. That can cause the cell to swell and lyse or burst. Hypertonic solution, the solute concentration outside the cell is higher than what is inside. Water will move then outside of the cell and cause it to to shrink. It says crenate. That's not something that you really know the term of, but that is the word for it. And it will lose water and eventually could lead to, like we said earlier, plasmolysis. That's overly shrinking the cell, and that can lead to cell death. So a little mnemonic that was created here for it. When I create these mnemonics, some of them I create myself, and if they are created by me, they're ridiculous. But if they're not created by me, they're created by ChatGPT. And this time we have I hope hyper cells to help you guys try to remember what is going on here. And let's see if I wrote down the explanation of that. Of course I didn't. Yes, I did. So isotonic, this is going to be no net movement of water. That's what the I is for, isotonic. H, hope, that's going to be hypotonic. Hope, hypo. The next one's hyper, and then the next one is cells. So that's not really, remember this is going on in movement across a cell membrane. And I guess that's how it's supposed to help you guys remember the 3 different terms for that, I guess. I ask it to do these things, and that's what it gives me. All right, so our example questions, which of the following best describes the movement of water in an isotonic solution? Okay, water moves into the cell, causing it to swell. Well, if it's isotonic, we should be talking about equal, right? Iso is referring to equal on both sides. So we're not just moving back into the cell. We should be kind of moving across both ways equally, nice stable movement. So it shouldn't be A. Water moves out of the cell to shrink it, right? So it shouldn't be B, because those are just one or the other. Water moves in and out of the cell at equal rates. It's going to be this one, because the D says water doesn't move across the membrane. The molecules never stop moving. So they're going to be moving at all times. That's just the way molecules are, y'all. They never stop moving. Even in solid state, they're moving. So it just is less, right? So water, it's not going to be D. You're always going to be moving. That's just basic chemistry. All right, next question. A patient is receiving intravenous fluids and the physician orders saline to be administered. The saline is labeled as 0.9% sodium chloride. Which type of tonicity is the solution, and why would we choose that for the patient? This might be a little harder if you aren't able to understand about the hypotonic and isotonic and things and the effects they might have on the cell. So we'll go through this. So A gives us the choice of hypotonic. So if we had this solution and it was hypotonic, would that be used to rehydrate the cells by causing water to enter them? So it could be used to rehydrate the cells to help water to enter them, but that's not the right answer. And I'll tell you why when we get to the right answer. The hypertonic, it's used to draw water out of the cells to reduce the cell's swelling. It would draw water out of the cells if the cells were swelling. And in this case, we're talking about our patient. And you can go into the hospital with any complaint whatsoever, and a lot of times get an IV. Are you dehydrated? Nope. So they're just trying to do what? Keep you hydrated. You might be a little dehydrated, yes. You might be a little overhydrated, yes. But this whole point of sodium chloride, 0.9% being given to you, is to keep you hydrated or bring you back into that, what is the word I'm trying to think about? I can't think of it right now. Oh, homeostasis. That's it. Homeostasis, where everything's supposed to be, copacetic, basically. So if we want to be copacetic and we want to get back to homeostasis, where everything's set and nice, then you want to give them isotonic solution. So that's our choices C and D. So we know it's not going to be A or B. It will maintain the balance of the fluids between the inside and outside of the cell. Or will isotonic cause cells to shrink by increasing solute concentration in the extracellular fluid? Well, no, it should be isotonic, right? Copacetic is what we're aiming for here. We want equal, we want homeostasis. So we're going for isotonic. So even if you are dehydrated and your cells are shrinking a little bit, or your cells are swollen a little bit because of whatever's going on in the cell body or whatever it is, either way, if you put cells in place in an isotonic solution, what should be isotonic to typical cells, they'll achieve that isotonic by adjusting to those slight different changes. If they were a little bit smaller, a little bit bigger, whatever it is, you put the isotonic solution in there to get those cells back to isotonicity. You don't try to compensate. That's why we give isotonic solution, 0.9% sodium chloride, whenever we give a saline solution to a patient. Okay. That's apparently it for chapter 9. The things that people get wrong. People do pretty well with these chapters. Chapter 25, the most missed concepts. It goes over the nitrogen fixation. And I feel like, because I looked over the questions for the exam, I really feel pretty strongly I don't have anything difficult about the nitrogen stuff on the exam. It might have just been like, what is the importance of, nitrogen in the soil or something? Because I don't go over the actual what nitrification is, what ammonification is, or what any of these things are. We didn't even talk about assimilation, but that's just the plants using it. But this is the breakdown of what those terms meant, where I mentioned briefly about nitrogen fixation. So that's going to be essentially turning that nitrogen that's in atmosphere into a form that plants can use. And then there's just different steps kind of in the middle or at the side, where we can be doing nitrification or ammonification, or denitrification, that these all can work together to make nitrogen usable and exchangeable within the soil environment, or really anywhere that we have bacteria dealing with nitrogen. So great. So this is what happens with nitrogen. And again, I'm not going to have you define any of these, but there are different ways that bacteria deal with nitrogen in the environment that is helpful for plants to be able to use it. I'm like 100% sure that's what the question is about. It definitely doesn't deal with the definitions, because I never ask about this because I think it's ridiculous, because I don't know it either. Okay. Like a couple of them and that's it. So that should put it into perspective, how I'm not going to ask you stuff that I don't know. It's stupid. So here we go. If you wanted to remember what all of them were, if you could remember nice ninjas always accept donuts, then that could help you remember nitrogen fixation, nitrification, assimilation, ammonification, and denitrification. Those are the terms that are associated with all of that. Again, assimilation is just dealing with how the plants are using it. All right, so anyways, which bacterial process in the nitrogen cycle converts ammonia? So these are harder questions than what you're ever going to need to know. But hey, which one would be converting ammonia into nitrite? And y'all, literally, I don't know the answer to this question, and I wrote it like a month ago, but I don't know. So I would have to look this up, and I'm not even kidding. So I would have to find, it's probably ammonification, I'm assuming. No, ammonia into NO2 would be this one, nitrification. Wow, so see, I would have to look at that. This is not going to be on your test. And this was me hoping that we would have kind of lectured over it a little more, which I have to lecture over this concept in order to remind myself about how it works. And I didn't lecture over it. And so we're not testing over it. But anyways, there's the answer for that. So agricultural practice, which bacteria is crucial for converting atmospheric nitrogen into a form that can be used by crops? And why would this be important for soil fertility? Again, I won't ask you about the processes. The second part of these might be important as far as introducing nitrogen into the soil that is usable by plants. That is literally what the question is going to be. And that's what the question was that was most missed on that exam, related to chapter 25 for that anyways. All right. And this is also chapter 25. They had previously had a question about the effects, basically, of, this is the effects of global warming, okay, on the environment and what effects it may have had. How can you imagine that global warming or climate change could affect, diseases and life on Earth as we're knowing it? Be aware of the general effects that it could have on the environment. I don't feel like this is stuff that isn't obvious. It should be relatively obvious. If you're increasing temperatures, then the further north you go, the more the mosquitoes are going to live. And that's probably the biggest concept behind this, because mosquitoes are heavy disease carriers, very heavy. If you think they only are just carrying like West Nile and or Zika, you are so wrong. I can't even tell you how wrong you are. These guys carry hella diseases. And the more mosquitoes that you have introduced, in fact, you've probably had mosquito-borne diseases and you just didn't know it. They carry so many freaking diseases, it's nuts. And they don't always present with symptoms or serious symptoms. So, but we want to keep the bad guys out, like malaria and dengue and chikungunya and things like that. That can be very dangerous. Zika is pretty bad. You know, if you didn't know, it can be very serious for unborn baby, leading to hydrocephalus and things like that. So it's bad, and microcephaly even. So it's bad. So you don't want those things to be happening as a result of these viruses that are carried by mosquitoes. So we want to control that as best we can. So higher temperatures put that at risk. Let's say habitat range. All right, that's just talking about mosquitoes again, y'all. Habitat range of mosquitoes will increase. Increased transmission rate of the diseases associated with mosquitoes. Altered disease dynamics. Global warming will disrupt seasonal patterns. If you can't tell, mosquitoes is the problem here. Okay. Hot temps allow disease populations. That's your mnemonic for this one, your memory device for this one. Hot temps, diseases. If it gets hotter, more diseases. That's a summary there. So how does global warming contribute to the increased risk of mosquito-borne diseases? So global warming, does it increase the breeding sites for mosquitoes due to less rainfall? That's not that. So we need rain and standing water for mosquitoes. So if there's less rainfall, that wouldn't be increasing the problem. But things that will increase the problem, let's see. Does it reduce mosquito populations by lowering temperatures? No, we have increasing temperatures. That's kind of the whole idea of global warming. It expands mosquito habitats and accelerates the development of mosquitoes and pathogens. This is sounding pretty accurate. And then D, it prevents replication of pathogens within mosquitoes. We wish, right? So C is our answer for that one. Not so bad. This is the type of question that you're more likely to face. I don't get crazy with chapter 25, y'all. I don't. I try to keep it simple. The only thing that I really feel like I needed to go over for chapter 25, even when I was like trying to, pare it down for you guys, was even the call of forums. Just saying. That's definitely on the exam. All right, which of the following, because I cut some stuff out, which of the following public health strategies is most likely to be affected by the expansion of mosquito habitats due to global warming? So reducing antibiotic prescriptions to prevent antibiotic resistance. I'm not sure what that would have to do with it. Enhancing vaccination programs for mosquito-borne diseases in newly affected regions. Okay, we would have to make a move to do that. If you have new diseases popping up, things like chikungunya, which is a real life disease, it's a potentially hemorrhagic fever-causing disease. It's a virus transmitted by mosquitoes. We do have it. They're now considering it to be endemic here in the United States. Chikungunya, that's the name of it. All right, not chickens. It's CHIKUNGUNYA. Spelling off the top of my head. Anyways, promoting low-fat diets. What? I mean, they should, right? Like, we want to decrease obesity-related diseases, but that doesn't have anything to do with global warming. And then increasing the use of personal protective equipment in non-endemic areas. I mean, if you have random, endemic means that the disease occurs there commonly, naturally. You expect it to be there. Do you want to increase the use of personal protective equipment in areas that are non-endemic? Not necessarily if you're not concerned about an active current outbreak, so an epidemic. If you're not concerned about that at the time, there's no need to increase your PPE to deal with that. So you really do want to go ahead if you think that new diseases are coming into your area, like chikungunya. There is a vaccine available for that. That if that's becoming an increasing problem, you're seeing more and more incidences, then you're going to want to vaccinate the population and get as many vaccines out as you can. That's exactly what we did with COVID. It may not have been with mosquitoes, but still, we had increased influx of a new virus that was extremely dangerous to our population. So we hustled hard to get those vaccines out. And before anybody wants to argue and try to say, yeah, we hustled so hard, we skipped regulatory procedures, they skipped the completion of one of the phases. That particular phase was going very well. They went ahead with the next phase, but they still completed that other phase. They just moved ahead before they completed it. So it did get completed, and it was looking good, and it was great, and it continued to look good even after they continued with the work on it. So no, they didn't cheat. They just did it, and it didn't go outside of regulations even. They still did it beforehand. All right, anyways, that's chapter 25. It's dealing more with conceptual either controlling things relating to global warming, making sure you have nitrogen available to your plants in the soil, or the coliforms, which you may have noticed didn't appear on here in our most missed questions. That's because people do pretty well with that concept, because they talk about it more, because it's definitely always on the exam. Coliforms, indicator bacteria. That's really all you need to know. All right, so chapter 10, our most missed concepts. We have the different parts of the enzymes. When you have a conjugated enzyme, oh geez. Conjugated enzymes. Simple enzymes are just protein. They don't have all this mess. Conjugated enzymes are the most common types of enzymes. The holoenzyme is going to be the whole functional enzyme. Sorry, my throat's getting dry. Okay, I had to get a soda. All right. The apo enzyme is what it says, a polypeptide chain. Okay, this is going to be the protein part, the actual like protein portion. And then you'll have the active site. This is where the substrate binds. Remember, the substrate is the thing you're acting on. Just the whole kind of deal. If you're trying to get from A to B as your product, then A is the substrate. All right, cofactors. This is any other non-protein thing that you need. So there's always non-protein. So it could be things like metal, or it could be organic, in which case it would be called a coenzyme. Organic doesn't mean protein, right? So vitamins are not proteins, but they are organic, so carbon, hydrogen based. All right, so this would include vitamins, NAD, FAD. These are going to help us transfer electrons, but they are coenzymes. They're carrying those electrons to interact with the apoenzyme portion of our oxidoreductase stuff that's going on later. Cool. So here, all apes can stay. Holo, so holo, apo, active, cofactor, substrate. These are all the possible parts you can have of any enzyme. So the active site, which would be this one here, and the substrate, these guys are involved in the simple. And the other, the holo, the apo, and the cofactors are for the conjugated. Well, conjugated also include the other two as well. So conjugated is all of them. Simple is only the stars. Okay, so that's our different parts of our enzymes. Remember, enzymes are just catalysts that are going to help chemical reactions overcome activation energy. Remember, from chemistry, a catalyst is just going to be helping that happen. They usually aren't going to be used up in that reaction. Enzymes are not used up, and they're not permanently changed in this reaction. They can be reused over and over again. They're just helping the reaction go. They're not giving up anything to the molecule, the substrate, to make it happen. They're just there to acetate. So, which of the following enzyme components is directly responsible for the chemical reaction by binding to the substrate? So, which part of our enzyme, when we're looking at, I already forgot the acronym, here all apes can stay. Let's just put that up here. Let's actually use it this time, right? All right. So it's asking us which of the following enzyme components is directly responsible for the chemical reaction by binding to the substrate. So we've got all of our components here. We've got the holo enzyme, which is the whole functional thing. We've got the apo enzyme. We've got the active site, which is where our substrate is going to bind. We've got our cofactor or coenzyme. Just put co. And then we've got our substrate. So if you can remember that those are all the parts of a functioning enzyme, now we can look back at this and be like, which of these parts is responsible for binding to the substrate? And then you can hopefully go through these, and you've been aware, hopefully as well, that you know what each of these kind of does, at least in a basic concept. So you could find that the active site should be the answer here, right? That's the one, that's the actual portion that's going to be binding to the substrate. I'll tell you right now that the active sites that bind with the substrates are always on the apo enzyme. They're always on the protein portion. So there's something there too, just FYI. All right, in the context of enzyme deficiencies leading to metabolic disorders, right, you have enzyme deficiencies, so low enzyme function due to metabolic disorder. Which component's absence or dysfunction is most likely to affect enzymatic activity and result in conditions like phenylketonuria? So this is our inability to process phenylalanine, an amino acid, and can cause some serious effects if it's not properly maintained. This is what they do the heel stick for. One of the reasons they do the heel stick for in infants whenever babies are born. So testing for this PKU. So this is a reduced enzyme activity from a genetic defect, and makes it so they can't deal with phenylalanine. Right. So in the context of enzyme deficiencies, which component is breaking down in this system? Which part of these guys is breaking down that's going to lead to our reduced enzyme activity? Could it be the active site? Then you would imagine that you would see almost none. Substrate, the substrate itself isn't going to be affected, right? The substrate itself is the thing you're acting on. Your genes aren't affecting that. The apo enzyme, this is the part that's going to be doing the actual protein work. So yes, potentially this one. And then D would be the cofactors. Again, this is the things helping the whole thing go, function properly. If for some reason you needed cofactors to work because you are a conjugated enzyme and you are no longer able to associate with those cofactors because of any genetic defect, that's still the apo enzyme that's having the problem, not the cofactor. So it's typically going to be the apo enzyme here that I would imagine would be the issue. It's the part that would be actually doing the work, helping overcome that activation energy in order to get there. Now, if you had issues at the active site, you could also have complete inability to deal with your substrate as well. So both of those could be possible, but it's more likely that if you're talking about the activity of that enzyme, that's going to be the action of the apo enzyme, because the active site is just where you're binding. The action done by the enzyme is going to be done by the apo enzyme portion. It may need a cofactor to accomplish that function. But anyways, let's see what we said here. Oh, good. I just said nothing about it. Yeah, it's C. It just doesn't say it directly. So yeah, it was following along the same line of thinking that I was getting at anyways. Oh, I made it really complicated. Apparently, when I wrote it, I didn't think about the active site could be affected too. But the APO enzyme is the one doing the action, so that's why it's the answer. You're not going to get a complicated question like that in the exam, but at least we talked through it in a complicated level so that we can maybe learn from it and make the other ones easier. I don't know. Anyways, I liked the first question better. So redox reactions is another part that people will be having problems with. The Leo says GER, or the oil rig thing. Everything occurs in pairs. We're going to always have one be the donor and one be the recipient. The electrons have to come from somewhere and then go somewhere. They always have to be accounted for. We talked about the electron transport chain, and it's the importance of electrons moving through in the electron transport chain, which makes it a series of redox reactions. And those redox reactions, what's going to create our proton motive force, which is that building up of the protons, the positives on the outside of the membrane, that'll let them come back in through ATP synthase to make ATP. And redox potential, this is just talking about the tendency of a molecule to want to gain or lose electrons. Okay, so in order to then remember all those, there's that old red enzymes always react. Yeah, I don't know what else to say about it. Okay, in a redox reaction, which term describes the process where a molecule gains electrons? Now, I can look at gains electrons and be like, oh, gains electrons, GER, that's the GER part. So then I'd be like, gains electron, that is reduced, right? If you need to write it down though, write it down. So this is reduction. All right, the next question. In the management of certain diseases, such as cystic fibrosis, medications that impact oxidative stress are used. What type of reaction is directly targeted by antioxidants used in these treatments? In case you aren't paying attention, it's going to be redox reactions. But we can look at these and say, okay, well, hydrolysis, they break down larger molecules into smaller ones. This isn't something that has to do with oxidative stress. And yes, oxidative stress does refer directly to oxidation. So we are talking about electron exchange here. Anytime you see these words in the context of science or medicine, probably anywhere else as well, that is what they're referring to at the core. So this is what it's talking about. So we know redox reactions are, reduction and oxidation reactions. That's what redox means. So this is electron transfer. So it's probably going to be this one. They involve the transfer of electrons between molecules. That's exactly what oxidative would be dealing with. Anyways, and when you're talking about antioxidants, when you're talking about being oxidized, if you want an antioxidant, you're trying to prevent your electrons from being stolen. That's what's going on. That's what an antioxidant is. So any antioxidant is preventing damage being done to any molecules in your cell by oxidative stress, which would be things potentially stealing away your electrons that you need for functioning of your cells. So that's basically what this is talking about here. Phosphorylation reactions, we're just going to knock off the others because we know it has to deal with electron exchange because it talks about oxidative, which is redox reactions. All right, do definitely know the difference between reduction and oxidation and be able to put them into play in a situation. That's a more difficult situation that I will give you on the exam, and I promise that. But I might test you. Electron transport chain in bacteria versus eukaryotes. This is usually, like I said previously when we were touching on what we talked about, this is usually referring to people not remembering that you don't have membrane-bound structures in a bacterial cell. Eukaryotes only have the membrane-bound structures inside. So that means, what I mean by that is bacteria don't have mitochondria. Okay. Eukaryotes, all of our electron transport chain is occurring on the cristae. or the folds of the mitochondria. Bacteria, this has to happen on the cytoplasmic, also known as the inner membrane. They don't have any other membrane for this to happen on. Literally, they don't have any other membrane. So this has to happen on their inner membrane. That's the electron transport chain only. The glycolysis and Krebs can happen in the cytoplasm for both of these guys. Okay, so that's what the question is on the test. It's going to refer to that, having to know that bacteria do it on their inner membrane and eukaryotes do it in mitochondria. All right, that's all this is getting at. So here it's trying to say, please make it more efficient. Let's see how it broke down that. Please, for the plasma membrane, which is the inner membrane, cytoplasmic membrane, that's for bacteria. Plasma membrane is a place for bacteria. Mitochondria is going to be the main location for eukaryotes. So if you remember, bacteria alphabetically comes before and evolutionarily comes before eukaryotes, then you could put those to work a little bit better. But there are environmental variations, I guess, depending on bacteria. And what I mean by that, environmental variations, that's talking about the aerobic versus the anaerobic guys. But man, I don't even worry about that. Okay, so our questions about the location of our electron transport chain. Where does the electron transport chain operate in bacterial cells? I could swear this is almost the question, like almost word for word. The inner mitochondrial membrane, the outer mitochondrial membrane, the plasma membrane, or the nucleus membrane. Break it down for you. It's asking us about bacteria. Bacteria do not have a mitochondria. They do not have a mitochondria, and they do not have a nucleus. Answer. Okay. Next question. In the treatment of mitochondrial disorders, such as Leber's hereditary optic neuropathy, which part of the cell is directly targeted by therapies aimed at improving cellular respiration. So you got a problem with cellular respiration, which is also aerobic respiration. They are interchangeable terms. And if you weren't sure, you could ask me. That would be fine. But respiration is always talking about this. Even when you're talking about breathing in and out in, physiology or something, you're talking about this, right? We already learned that. So respiration, in this case, on the cellular level. So that's going to be creating ATP through glycolysis, Krebs, and the electron transport chain. And we're going to look at which part of the cell we might want to look at if we're having issues with your cellular respiration or aerobic respiration, right? Right. We are human beings, and it also even says mitochondrial disorders up here. So we're human beings, so we're going to have mitochondria, and that's where our electron transport chain takes place. Glycolysis and Krebs, again, in the cytoplasm. So the plasma membrane is not where this happens for us. That's where it happens for bacteria. The inner mitochondrial membrane, that is correct. Remember, mitochondria have two membranes. They have the outer surrounding membrane like this, and then they have an inner membrane. And their inner membrane looks like this. This big old membrane all the way around it. But then we have electron transport chains all along these kind of folds or whatever, so you can have a whole bunch of them shoved into one mitochondria. So there's all your cytochromes or whatever at each of those spots. So that's our inner membrane, and that is equivalent to the plasma membrane of bacteria, maybe specifically because we've got two membranes, gram-negative bacteria, right? So you can see that that could be the resemblance. We're just squiggling that inner membrane around. So it's our inner membrane of the mitochondrial membrane. The endoplasmic reticulum is not involved in cellular respiration, and neither is the Golgi apparatus. Remember, the ER is where we're going to see protein and other molecule production and modification. And the Golgi apparatus is for like packaging and transport. Just to keep us up on the knowledge with that. All right, so next we have the proton motive force. The proton motive force is a term referring to that buildup of the pluses, the hydrogen ions, outside of our membrane. We create that gradient, and that gradient itself with a positive on the outside and it having negative, therefore, on the inside, respectively, that is what drives ATP production. And you need that. You need that gradient at all times to make ATP happen, okay? When we're shooting our pluses, our H pluses, our protons out of the cell to create that gradient while we're moving our electrons through the electron transport chain, that is chemiosmosis, all right? So chemiosmosis is the movement and creation of that gradient and the ability of that gradient to want to come back in and create ATP. So chemiosmosis is moving the H pluses out and then making them want to come back in. All right, energy storage and utilization is basically achieved by creating that gradient. It's like potential energy. It wants to come back in, just like water behind a dam. It's going to want to equal out. And bacteria, proton is generated across the plasma membrane. Okay, we already talked about this. Be aware of where the, if I were to ask you, where's the proton motive force generated? You should be able to tell me that in bacteria, the proton motive force is generated across and outside of that cytoplasmic or plasma membrane, the inner membrane, right? And that in the eukaryotic mitochondria, they're going to be creating that gradient on the inner mitochondrial membrane where we had that whole folds and everything like that. The cristae is what it's called. Okay. Now we're in chloroplast right now. All right. It says, please all enzymes calculate. Let's see what it means by this. If I wrote it down, I did. So the purpose, the main function of the proton motive force, is drive ATP. It says ATP. So I would even say proton ATP. I am assuming, so enzymes calculate. So enzymes involved in energy creation. Energy calculate is cell activities, apparently. So please all enzymes calculate. Protons. Protons are used to make ATP, energy, and drive cell activities. That's what that's talking about. Great. If you want to use those, I don't know if that's at all useful for anybody ever. Some of them are more useful than others. All right, what is the primary role of proton motive force in cells? We're creating a H plus gradient to drive ATP synthesis, right? So like a dam, it's literally like a dam. So with hydroelectric dams, you are holding back water and letting a little bit through to cause like turbines to spin. And the spinning of those turbines can create electricity from that. So we're doing exactly that. We are letting through a little bit of H pluses from the H plus dam that we've created behind us and using that to spin our turbine of our ATP synthase to drive production of ATP. Okay, so that's what's happening with our proton motive force. So here we know we're going to go straight to our energy, just like a dam, we're making energy, ATP synthase. So in the treatment of mitochondrial diseases, go figure, we're talking about mitochondrial, such as mitochondrial myopathy, which cellular process is directly impacted by therapies aimed at enhancing the proton motive force. So they're trying to create more protons behind the dam to help drive production of more energy in these diseases that occur within these affected people's mitochondria. So anyways, this is going to have to be ATP production. Mitochondria, that's the entire purpose of the mitochondria, is energy production. Only ATP, energy production. It's just the whole point. So it's not going to be anything else. We're not going to target anything else. Proton motive force is involved in making ATP. ATP is made in your mitochondria. Alrighty, final electron acceptor. I don't know how people keep getting this one wrong. This one's kind of frustrating on me as far as like when I look at these and I see that people are getting it wrong still, but it is one of the most commonly missed concepts. The final electron acceptor in aerobic respiration is, I feel, literally in the word. Okay, what is the final electron acceptor in aerobic respiration? It's oxygen. It's oxygen. Yeah, we talked about like aerobic respiration and anaerobic respiration and fermentation. What's the difference in all of them? What does aerobic have? Oxygen, right? What does anaerobic have? Not oxygen. And why does fermentation happen? Not oxygen is there, right? No oxygen available. So aerobic respiration is defined by the oxygen being there at the end. It is not ever going to be aerobic respiration if you're making ATP without oxygen. You have to have it. It's why you suffocate. So the final electron acceptor, the whole end of it, you've moved your electrons through your little cytochromes to make your H plus gradient, and their H pluses are coming back in. You got to get rid of the H pluses, but you also got to get rid of those electrons because those electrons will also build up at that chain, and then you can't move them through to pump more hydrogens across. So we got to get rid of both of them, the negative and the positive byproducts that happened. The negative being the electrons, the positive being the H pluses. We get rid of both of them in the same exact place at the same exact time. And that is by splitting an oxygen molecule to make H2O as our product. Oxygen is the final electron acceptor. That electron is going to go into water when we split that oxygen up, right? We're shoving it into that water molecule, and then you're going to pee it out. Congratulations. Okay. So you have to have oxygen there. If you don't have it, you're not going to be able to maintain your gradient. If you can't maintain your gradient of your H pluses, you can't move them through. Additionally, you have to have oxygen there because you have to get rid of those excess electrons, because the same thing, but instead of the gradient outside coming in, we're just talking about shuttling the electrons through the cytochromes. You got to be able to shuttle them. If you got too many building up, you ain't going to shuttle. So that's the problem there. You got to get rid of both of them. Got to get rid of electrons. You got to get rid of H pluses. Turns out when you combine these two together, you just make a regular old H that's ready to share its electron with somebody. And you can do that twice. And then you can share it with half of your O2 molecule for just one oxygen here. That's what's happening. Okay. H pluses, by the way, they don't share electrons. Why do H pluses not share their electrons with things? Because they don't have any to share. Okay. They have to have electrons to share electrons. All right. So this is what's happening. This literally, the question will be like, what is the final electron acceptor in aerobic respiration? And people are getting it wrong. Please don't be the people. Okay. Because it's one of the most annoying things to me. Because I feel like it says it in the word. But this is why, okay? So you can see aerobic, you should automatically be thinking, this is respiration. Aero means with oxygen at the end. You need oxygen for this one. Okay, so there's also anaero. What does it need at the end? Not really sure, but it's not aero. It's not aero, so it can't be oxygen. That should answer the question for you. That's the difference. So there's a reason I would bring it up. Anyways, oxygen accepts electrons willingly. Oxygen accepts electrons willingly. Oxygen loves sharing electrons with other organisms. Well, oxygen loves sharing electrons with other atoms because it can hog their electrons. Y'all are just so good at hogging the electrons. So it'll be like, yeah, sure. I'm happy to join in. Yeah. Where do I stand? That's what oxygen's saying, because it cannot wait to hog those electrons. It is just so excited to hog those electrons so we can have that O be slightly negative and the H's be slightly positive so we can create our interactions with our polarity. That's what drives a lot of this stuff, right? So it is so happy to do this. All right. What is the final electron acceptor in aerobic respiration? And what does it form as a result? Well, it gives the answer away. All you would need to know on this one would be oxygen. You know your final electron acceptor for aerobic has to be oxygen, right? So our final electron acceptor in all of this whole BS that we went through, it always ends at oxygen. And we talked about that in class. And yes, it forms water. That's our byproduct because we're getting rid of those H pluses, those E negatives, and the half of the O2 will be spared in the middle of that, right, to make H2O. So great. So we can get that in our minds of how that's all working together. But the most commonly chosen answer by people I'm imagining that do not study well enough, they just see words that were associated with things that happened at the same time. They choose carbon dioxide. Carbon dioxide is not an electron acceptor in any of this. It's a waste product. And it's not, so that's the thing, right? It's not accepting electrons for us. We're not bringing in carbon dioxide to accept electrons for anything ever. It's a waste product. It's gone. Like it's not doing anything for us. That's why we exhale it. Oxygen, though, we're bringing it in specifically to accept electrons. Is the literal reason that you breathe in. You breathe in literally only for those oxygen molecules that you're breathing in to accept electrons. That's the only thing it's doing. Please remember at least that. If you take this, if you get out of metabolism, please remember at least that. Why are you breathing in and why are you exhaling? Because I feel like if you're going to go into healthcare where you're going to be looking at people's respiration rate, you're going to be looking at, their carbon dioxide output, and you're going to be looking at these things, you should at least be able to tell me where that carbon dioxide comes from and why you're breathing in oxygen. And it shouldn't be because you inhale and exhale. I'm just saying. If you take anything out of this, take those basics away with you. Okay. So, and if you're going into if you're going into be a medical doctor or PA, or you want to be an LPN or something like this, then you really need to know this because you'll have to know this at the actual molecular level. This is baby stuff. So anyways, in patients with conditions that impair oxygen utilization, so they can breathe in just fine. It's just they're not able to use the oxygen for its intended purpose. Now, like I just did in my monologue, my villain monologue over there, I just told you guys the only thing you're using oxygen for in your entire body ever, you're using oxygen as your final electron acceptor in literally the electron transport chain, but you could even look at it overall big picture in aerobic respiration. But the literal place that it's happening, of course, is going to be at the very, very, very end, which if you're thinking about the very, very, very end, because it's the final electron acceptor of aerobic respiration, we're not talking about glycolysis then, right? That's not the end. We're not talking about the Krebs cycle. That's not the end. What's the end of aerobic respiration when you're making ATP? The electron transport chain. So at the end of the electron transport chain, you will use oxygen as our final electron acceptor, right? So this person can't do that. They can't use the oxygen as their final electron acceptor very well. So this would be, for example, COPD. So their actual cellular respiration is being affected because of this. So which aspect of it is being affected? Are we inhibiting synthesis of glucose from pyruvate? That's not even a thing, by the way. Pyruvate, right? We make two of those from one glucose in which part of the cell cycle, which part of the aerobic respiration? That's going to be glycolysis. But this isn't even that. This is the opposite. So definitely no. Is it relating to the transfer of the electrons through the electron transport chain? We'll come back to that. Is it the reduction of oxygen? Okay, so what's reduction? Gaining electrons, okay, to form carbon dioxide. Or is it the production of ATP in the cytoplasm? So we're not talking about production of ATP in the cytoplasm because our ATP isn't made in the cytoplasm, is it? Where's our ATP made again? The mitochondria. No, not that. All right, so if we're looking at these last two, right? Let's think of a better worded version from what I'm gonna know I didn't write down notes for it. Okay, so my choice is B, the transfer of electrons through the electron transport chain. Why would that be affected? If we cannot get rid of those electrons with our final electron acceptor, right, which we just wrote down below this, oxygen is our final electron acceptor, right? Now, if oxygen is gaining those electrons during that process, it's accepting those electrons. The same thing as gaining. Gain electron is reduce. So we are reducing oxygen in this process. This talks about reducing oxygen. Are we reducing our oxygen to form carbon dioxide? Is that a thing? Could that be one of the answers? If you've been paying attention, I hope you have, then I would hope that you would know that it can't be C, because we are not reducing oxygen to form carbon dioxide. Does your carbon dioxide come from the oxygen that you breathe in? No. Where does your carbon dioxide come from? From breaking down your glucose molecules to steal the electrons from it. That happens in the Krebs cycle mostly. So you give off carbon dioxide in the Krebs cycle, breaking down, stealing carbons off what used to be your glucose, which is your food, not the oxygen that you breathe in. That doesn't even touch the oxygen that you breathe in. So this cannot be the answer. Your carbon dioxide doesn't come from that. No, you need to understand these processes at least well enough that you can look at the chart that I drew earlier on in the little review preview portion, which was pretty good rundown of the basics of what you're making, when, and why. If you didn't see that, then I would say go back to that and look at it when you have access to the recording, because I can't apparently keep it on the slides. So anyways, it's going to be B, because you need to get rid of those electrons as they move through, so you can keep other ones moving through next, and other ones moving through next, and other one moving through next. So oxygen keeps that going. That's why that's the answer. So I hoped that there was an easier, less wordy version of that. I can't believe there's another one. I thought it was done. Apparently people don't do very well in chapter 10. Okay, so which part of aerobic respiration makes the most ATP? I know I went over that in the beginning of the little review thing, but here's the breakdown of literally what is made at each step. And I almost feel like I should almost go back through it again one more time, but I feel like maybe I'll like draw it out or something and just like post it online for you guys to see so that you have it available like permanently. I'll see if I can do that for you. But anyways, glycolysis makes a little bit of ATP. Yeah, it does. It makes a little bit of ATP. But what's the big daddy result of glycolysis? Pyruvate. So here it calls it the link reaction, pyruvate to that acetyl-CoA. Doesn't make ATP. Okay, it doesn't do it. All right, next we have the Krebs cycle. So glycolysis makes pyruvate. We do make a little ATP in the process too. So we go through the Krebs cycle. Whole point is to take what's left of your pyruvate and shuttle it through and steal electrons off of it in the meantime. We'll kick off some carbon dioxide and we'll kick off some ATP in the process too. Might as well, too. Next, we have the electron transport chain and oxidative phosphorylation. Oxidative phosphorylation is the process that's making the ATP. We're moving electrons through things, and it's oxidative because of how we're moving the electrons through. So oxidative phosphorylation, we're phosphorylating, adding a phosphate group onto ADP to make it into ATP. So that's the whole point of the electron transport chain. Anyways, that's the whole point of the electron transport chain. I guess I should say that again. We're making ATP. If that's the whole point of the electron transport chain, you better believe, I mean, that's the whole point that it exists. I told you glycolysis exists to make pyruvate. I told you that Krebs exists to make electron carriers. Those electron carriers are going to go through the electron transport chain to make ATP. So guess where you're going to make the most ATP? Big daddy time, y'all. It's going to be 28 to 34 ATP as opposed to 2, as opposed to like 2 or whatever. So whatever. I'm not going to go through all the details of how much you use up in which step either, because there's stuff like that going on too. But you don't use up any in the electron transport chain. You're only making. So great people create energy. It's telling us that's great people create energy. This is great means glucose. People is referring to pyruvate. It's like the middle reaction. Create, I guess, is the cycle, the Krebs cycle or citric acid cycle. And energy, where the energy is being made, the electron transport chain, ATP, that's where it's going to be at. So how many ATP molecules are produced directly during glycolysis from one molecule of glucose? And y'all are probably sitting there being like, you just said it was 2. And I'm not going to test you over the number anyway. It doesn't matter. But it makes 4. It's just 2 are used up. That's all. So it's 2 net. So it's talking about directly made. You do make 4, but you're going to use 2 of them up right away. In patients with a condition that affects mitochondrial function, so energy making, such as mitochondrial myopathy, so we're dealing with a muscle disorder that occurs because of your mitochondria not making energy properly. Which stage of aerobic respiration, so we already know O2 is involved, is most directly impacted in terms of ATP production? Is this going to be something that affects the glycolysis the most, the pyruvate oxidation, so that link reaction the most, the citric acid cycle or the Krebs cycle the most, or the electron transport chain? So if you're having a real issue making enough ATP, it's going to be the electron transport chain, because you're going to be barely affected if you're having issues making ATP in the others. It's got to be D. Oh my Jesus. Okay, so photosynthesis has light reactions and it has dark reactions. Here's a breakdown of what is exactly going on in our light reaction. Light acts wonders producing oxygen. So really, honestly, the light reaction is using energy from sunlight to excite pigment molecules. That's going to drive essentially the exact same thing that we just saw, creating H pluses, gradient, shooting through the, I'm trying to say, shooting them through ATP synthase, right? Okay, so that's great. So we're going to be doing that. We make the little bit of ATP. Then we're going to go into the Calvin cycle, which is the light independent reaction. That is where we're going to be fixing carbon dioxide into our 6 carbon molecules like glucose. We needed that ATP from the light reaction to do that. So that's the summary of what's going to be happening on with this. What I want to say about this. Just don't want to miss something that might be important. Okay. So what is the primary function of the light reaction in photosynthesis? We're trying to make ATP, energy. The light is just making energy. We're not doing anything with carbon dioxide Sorry, it just kicked me out when it did that. Does anybody have any questions? I know that was long. Okay, of course, you guys know you can always email me if you have any questions. I'll try to get that little summary drawing guy so you guys know like pretty much what you need to know off of all the aerobic respiration, and I'll post that in the announcements. Otherwise, I guess we'll go and I'll post this whenever it processes. Thanks for joining in.